Welcome to the Avidity corporate presentation. My name is Sarah Boyce. I'm a Chief Executive Officer at Avidity, and I'm joined today by my colleagues Joe Boraldi, who is our Chief Operating Officer, Mike McLean, our Chief Financial Officer, and Art Levin, our Chief Scientific Officer. During the course of this presentation, we will be making forward-looking statements. This slide gives you the 30,000-foot strategic overview of the company. We believe that we have pioneered and created what is a new class of therapeutics, the antibody oligonucleotide conjugates. Our goal is to be able to take oligonucleotide therapies and deliver them to a wide range of serious diseases. The platform combines the selectivity of a monoclonal antibody along with the precision of an oligonucleotide, the goal being to access previously undruggable tissues and cell types. Oligonucleotides as a class of drug have generated a great deal of value for both patients and for investors, but really through an approach of only targeting parenchymal cells in the liver or delivering via an intrathecal injection. What we're looking to do through our technology is to be able to expand the sphere, so to speak, for oligonucleotide therapies. We have a very large focus on muscle diseases where we have 5 different programs, and we'll go through those programs in turn during the course of the presentation. AOC1001 is our lead product candidate for myotonic dystrophy. We're expecting an IND next year and starting the first clinical studies next year as well. Myotonic dystrophy is a devastating rare disease for which there are currently no approved treatments. And it's our goal that those first clinical studies actually start in patients, given both the nature and severity of the disease as well as the level of unmet medical need. An example of where we're beginning to expand the technology is our strategic collaboration with Lilly, which is around a specific number of immunology targets. We work in collaboration with Lilly on those programs, and we also have our own programs in immunology that we can work on as well. What this has enabled us to do is to be able to accelerate the technology into a therapeutic area faster than we otherwise would be able to do on our own. Last year we completed our Series C where we raised $100 million, and that round was led by RTW, and Rod Wong, as part of that round, also joined our board. As a company leadership team, we actually have a lot of experience in the oligonucleotide space. Myself, of the 7 approved and now commercially available oligonucleotide therapies, I led 2 of those programs and was very closely involved in a third, and have spent my career building global companies, in particular in the rare disease space, and taking drugs through from development through to late-stage development, approval, and ultimately commercialization. With that, I'll ask Mike to introduce himself, and then Joe and Art. Mike, over to you.
Thanks, Sarah. I actually joined the company as CFO approximately 3 weeks ago, and prior to that, I worked with Sarah at Axia Therapeutics, where we were responsible for a pipeline of products and product candidates of approximately 5 oligonucleotides, which we launched 2 commercially in multiple markets around the world. Prior to that, I was CFO of PureTech Health, and I actually cut my teeth on coming into the biotech industry when I joined Biogen approximately 15 years ago as their controller, increasing my responsibilities until I ran the global financial infrastructure. And with that, I'll turn it over to Joe.
Thanks, Mike. I'm Joe Boraldi, Chief Operating Officer. Like Mike, I'm a relatively new addition to the management team, having started in February. However, I do have considerable amount of experience in oligonucleotide therapeutics, having spent 15 of the last 20 years of my career in the space. I actually started as a bench scientist doing drug discovery early in my career. However, more recently, 11 years ago, I started at Ionis Pharmaceuticals on their business development team. However, when I left in January, I was responsible for running all of business development, alliance management, and competitive intelligence. So I've participated on every one of their transactions over the last decade, bringing in roughly $2 billion in upfront money in that time.
And my name is Art Levin, and I'm responsible for research and development. I have 25 years of oligonucleotide experience. I've been at Avidity since its inception, and what you're seeing here is really the outgrowth of that work. I've been in oligonucleotide development. I was the senior vice president of drug development at ISIS Pharmaceuticals. I ran the development organization as the chief development officer at Santeris Pharma before they were taken over by Roche, and I ran research and development at Miragen Therapeutics. In 2013, I got a call from Troy Wilson, our founding CEO, saying they had a novel way of developing oligonucleotide therapeutics. We've essentially taken that technology, adapted from it, and have created something that's entirely new here at Avidity. Sarah?
Thanks, Art. Now moving to slide 6, I'm starting to look at our portfolio and in particular our programs in muscle disease. In each case, where we are able to achieve a great deal of synergy from one program to another is the fact that we're conjugating the relevant oligonucleotide to the same monoclonal antibody. In this case, it's a monoclonal antibody targeting the transferrin 1 receptor. So the antibody is the same, the linker is the same, but the relevant oligo is different relative to each disease. I have a couple of comments from a strategic perspective on this slide. One of the things that you'll see is we have 4 programs in the rare disease space. Rare disease programs are the types of programs that we plan to develop and ultimately commercialize ourselves. The program that doesn't quite look like the others on this slide is our program in muscle atrophy. Muscle atrophy as an indication can potentially be something that is in the rare disease space and also something for very broad populations, for example, like cancer cachexia. In the case of muscle atrophy, if we were going after that sort of broad indication, that's where we'd like to commercialize and bring in that additional resource from a large pharma. So this also speaks to strategically the types of programs that we plan to develop and commercialize ourselves, as well as our sort of partnership strategy for much larger indications. We're going to go through in each of these programs in turn as we dig into the presentation. So what I'll do now is move to an overview of our platform and technology. So an AOC is simply as described. We are conjugating an oligonucleotide to a monoclonal antibody using the specificity of the antibody to be able to deliver the oligonucleotide to the target cell. It's a really simple concept, but as with many simple ideas, they're actually more complex when it comes to actually working them out and bringing them to fruition. In terms of from a development of technology perspective, every aspect of this mattered, both in terms of from the antibody itself to the linkers, to the oligochemistry, to the arrangement in space, and in terms of those inventive steps in developing the technology, that also allowed us to build our IP estate around it. Now, looking at the potential breadth of the technology and where we're looking to move forward, if we go to slide 9, we're not limited as to the type of oligo that we can conjugate to an antibody. For example, in our DMD program, where we're looking to achieve exon skipping, we're using a PMO approach. In all our other programs, we're using an siRNA approach for the simple reason that we believe it's the better of the approaches And we'll go in and show you data as to why that is the case. One of the other ways to also look at the potential breadth of technology is as we look at slide 10. So remember, in terms of we're not limited to the type of oligo that we're conjugating to the antibody, nor are we limited to the antibody itself. So we can conjugate the relevant oligonucleotide to a different type of antibody in order to achieve targeting of different types of cells. So on this slide, what you're seeing is a range of different experiments shown in one place. When you see the different colors in the bars, that's in relation where we're conjugating our oligo to a different antibody. The engineering work around the technology was conducted in the liver, but that's not where we're looking to target our technology, because there are several companies that have very successfully delivered oligonucleotides to the liver. What we're looking to do is go where everybody else has been unable to, and that's in looking at things like skeletal muscle, cardiac muscle, immune cells. Where you see the moss green in the middle of the chart, that's where we're conjugating to the antibody targeting the transferrin 1 receptor. And then as you move across, for example, as we look at macrophages, that's where we're conjugating an oligonucleotide, in that case to an antibody, to a receptor that is specifically expressed on those cells. From a strategic perspective, it's our goal over time, as we identify more of those antibody receptor pairs, to be able to expand the numbers of different cells and tissue types that we're able to target with our technology. So moving to slide 11, that gives you sort of the overview of what we're trying to achieve. So the first aspect is looking to be able to address previously undruggable tissues and cell types with oligonucleotide therapies. They include things like muscle cells and immune cells, but also many others. One of the other aspects is we're looking to mitigate the toxicity associated with oligonucleotides, and that's through a range of mechanisms, one being the selection of the most potent oligotype, but also a very targeted delivery approach through the conjugation to a monoclonal antibody, as well as our goal to achieve relatively infrequent dosing. Art will show you during the course of the presentation our data where we're able to achieve target knockdown 12 weeks out after a single dose in monkeys. And it's our goal as we look to ultimately to be able to deliver once every 3 months. One of the other advantages of the platform is the aspect of reproducibility and scale. In particular, in the fact that we're using the single monoclonal antibody in the case of our muscle program, as well as the same linker. So that allows us to achieve a great deal of synergy, for example, from a CMC perspective. Now we're going to move into more details around each of our individual programs and actually show you some of our data as we get into it. We'll focus the majority of the time on our lead program in myotonic dystrophy, but as well as also sharing with you the exciting data from our other programs as well. And with that, I'll hand over to Art to start with myotonic dystrophy.
This is Art Levin, and what you're looking at on slide 13 is a brief description of the disease myotonic dystrophy type 1. There are about 40,000 people in the U.S. that are thought to have the disease, although this may be an underestimate since there are no treatments at this point in time. There are no approved therapies. It is a monogenic autosomal dominant disorder. It leads to progressive muscle wasting. It also leads to cardiac conduction defects, and of course associated with muscle wasting is the fact that these patients ultimately lose the ability to both be ambulatory as well as even as functions as simple as breathing. The mechanism of the disease is well understood. It's related to a mutation in a gene called DMPK. And on slide 14, we have a cartoon of the disease process. In a disease process, there are a number of, you can see on the left-hand part of the slide, you can see in the gene called DMPK, there are a number of CUG repeats. Normal subjects have 25 CUG repeats or less. Subjects with myotonic dystrophy can have many, many increased numbers of CUG repeats, up to thousands. Those CUG repeats in the messenger RNA for DMPK actually result in the concept that these CUGs are self-complementary. They form hairpin loops in the nucleus, and those hairpin loops, which are highly enriched for CUGs, attract a CUG binding protein called muscle blind-like protein, which is abbreviated here MBNL. If MBNL is being sequestered by these CUG repeats, the MBNL that's required for downstream function, for its normal function, is missing. And the normal downstream function of MBNL is to splice and help process other messenger RNAs. So the disease that we're talking about here is actually related to the missplicing or the misprocessing of a number of different proteins. And there's a chloride channel in skeletal muscle and a calcium channel in cardiac muscle, which are known to be misspliced. In fact, there are dozens of proteins that are known to be misprocessed when muscle blind-like protein is sequestered on these CUG repeats. A therapeutic rationale that we have is that if you knock down the expression of DMPK, you're going to reduce the number of CUGs that are trapped in the nucleus. You're going to free up muscle blind-like protein from its sequestration associated with the mutant CUG repeats, and muscle blind-like protein can perform its normal function. In fact, what one would expect with this disease is that as muscle blind-like protein is freed up, it can then begin to properly splice proteins, and then over time, as proteins turn over, the improperly spliced proteins will be exchanged for properly spliced proteins. The fact that this mechanism can actually work has been demonstrated in a clinical trial that was done by Biogen Ionis, a phase 2 trial. In that trial, they're giving doses of up to 600 milligrams as a loading regimen, and then weekly maintenance regimen after that. The high dose essentially gave 600 milligrams 8 times over the course of 6 weeks, 4.8 grams of drug over that period of time. And what they found was that in 2 of 6 subjects or patients in that trial, they saw actually a change in the splicing patterns that all moved in the right direction for 22 different known biomarkers for splicing defects in myotonic dystrophy. The issue with that dose of drug is that it's extraordinarily high. Non-clinical studies in normal non-human primates demonstrated that they need about a concentration of about 1 micromolar in order to produce a 50% knockdown of the expression of DMPK. The concentration that they achieved in this clinical trial was about half a micromolar. So they only achieved 50% of the drug that they wanted in the muscles of these patients. And of course, a dose of 600 milligrams of an phosphorothioate antisense oligonucleotide have been associated with some pretty severe toxicities in the past. And so Biogen Ionis halted the trial, didn't dose escalate further than 600. The data that I would like to show you is some data that we recently generated, and that's on slide 15. We're now taking an siRNA rather than a naked antisense molecule that was used in the previous studies. We're taking an siRNA at a dose of 2 milligrams per kilogram. We're conjugating it to a monoclonal antibody to the transferrin receptor 1. The transferrin receptor 1 will bind to the outside of a skeletal muscle cell. It will then be internalized. In the process of that internalization, the antibody is ultimately degraded, and the siRNA can be released and produce its normal function. And in this case, the normal function of this siRNA is to knock down the expression of the DMPK RNA. So the axes that you're looking at here is expression on the left-hand side and time over the course of weeks on the x-axis, and you can see that a single dose of 2 milligrams per kilogram produced 75% knockdown that was essentially continued through 12 weeks without any evidence of recovery. The concept that people generally ask at this point is how much knockdown do you need in order to get therapeutic activity? And although the number isn't exactly known at this point, we do know that if two patients present with 50% different numbers in CUGs, they will have a phenotypically different disease phenotype. The more CUGs, the earlier the onset, the more CUGs, the more severe the nature of the disease. So we're shooting for a a dose which will produce approximately a 50% reduction in DMPK levels. Another question that comes up frequently is the fact that what happens if you knock down DMPK? Are there toxic sequelae associated with the loss of DMPK function? And to our understanding, that's not the case. There are homozygous null mice for DMPK. They don't show any phenotype. And more recently, genome-wide analyses of looking for natural mutations and loss of function have not been associated with disease states. On slide 16 is a key slide because we demonstrate here that we can knock down the expression of DMPK in tissues which are important for the disease. So what you're looking at on the left-hand side, again, is the change in gene expression versus the dose of siRNA. This is a dose response curve done in non-human primates. The dose in all cases that I'm showing you here were administered intravenously. And what you can see is that a dose of 1 milligram per kilogram, 6 weeks after administration, still produces a 50% knockdown in DMPK. And if you go as high as a dose of 5 milligrams per kilogram, you can see that we're getting into the range of almost 88% knockdown. At week 12, after a single dose of 2 milligrams per kilogram, on the right-hand side of the slide, what you can see is that all the way out at week 12, there's still a 50% reduction in cardiac DMPK and almost a 75% knockdown in the expression of DMPK in the diaphragm. So consider the concept that we now have a drug which is long-acting. We now have a mechanism of action that is going to allow us over time to replace misspliced proteins with properly spliced proteins. So we have the ability to potentially not only slow the progression of the disease, but to actually increase the functionality of some of the lesser affected muscles, allowing us to actually have a positive effect and a positive change on the phenotypic changes in patients with the disease. Also note, as shown on the last slide and shown on the left-hand panel on this side, we have significant activity after a quarter after the initial administration, which is leading us to project that we will be able to dose this drug relatively infrequently in patients, perhaps even as infrequently as once every quarter or maybe even less. This is completely analogous to what the Medicines Company and Alnylam are doing with inclisiran, where they're using an siRNA inhibitor or PCSK9, and they're getting good activity when they dose that particular drug twice a year. What I'm showing on slide 17 is that if you look at most skeletal muscles, and here there's a large collection of skeletal muscles that are shown, each of those bars represents a different skeletal muscle. The green panels represent a dose of 6 milligrams per kilogram 4 weeks after administration. The blue bars represent a dose of 2 milligrams per kilogram 12 weeks after administration. And the key point here is that after a dose of either 6 or 2 or 6 milligrams per kilogram, what you can see here is that we get, at the high dose, 6, we're getting greater than almost 80% knockdown over the course of the study, and this is at 4 weeks. And then below the bars, what I'd like you to look at is the concentration of drug that we've measured in the tissue. And the concentration of drug here is expressed as nanomolar. Recall that in the Biogen Ionis study, they were trying to achieve micromolar concentrations. Here we're almost 3 orders of magnitude more potent than was that phosphorothioate oligonucleotide. And there are lots of reasons for that. But another point that should be raised here is that on the right-hand side of the figure, what you can see is that you're still getting significant knockdown, even when the tissue concentrations in the muscles, those individual muscle biopsies, is in the sub-nanomolar range. So this represents the efficiency of loading RISC, the RNA-inducible silencing complex, and allowing, and once RISC is loaded, you get great interactions and a great deal of knockdown. What I'd like to address is a point that's been raised by some who are thinking in terms of old dogma. So there's some very old dogma in the siRNA world that there is not siRNA mechanisms only take place in the cytoplasm. And that's not the case at all. In fact, David Corey at UT Southwestern, probably a decade or more ago, demonstrated that all the components for siRNA activity exist in the nuclear fraction as well as in the cytoplasm. And because the important disease-related messenger RNA here, the mutant form of DMPK, is trapped in the nucleus, we thought it was important for us to be able to demonstrate that, in fact, we get knockdown of the nuclear localized DMPK. So in this experiment, what you're looking at again on the vertical axis is the expression of the MPK, and what you can see is that control cells don't have any knockdown. In the green bars, either you can see that there's significant knockdown in either the cytoplasmic fraction or the nuclear fraction or in the whole cell extract. So the concept that siRNA is inactive in the nucleus is simply old dogma. What we've done here is we've simply outlined the steps that we've taken and where we've gone with our program at this point in time. We've demonstrated great reduction of target messenger RNAs. We've demonstrated, although it's not in this presentation, improvements in biomarkers. We've been able to demonstrate that we're much more potent than unconjugated oligonucleotides, long duration of action, allowing us to propose potentially quarterly dosing or maybe even longer, and the effective delivery of an siRNA, and the efficiency of which siRNA knocks down a target. Sarah has already pointed out that there are numbers of years of experience of synthesizing monoclonal antibodies. We're building on 30 years of experience synthesizing monoclonal antibodies. We are building on 25 years of oligonucleotide synthesis experience at the commercial level. And of course, we're building on the experience that's been gained more recently with ADCs. Our first clinical trial is expected to begin in 2021. There are some players in this field. So there is some competition. We think the important competition that's actually targeting the toxic messenger RNA are being approached by Audentes and Dyne. Audentes is taking an approach where they're using an AAV vector to deliver a vectorized antisense molecule. So this technology will suffer some of the same pains with all AAV vector technology, as well as definitely having to do some work to make sure that a vectorized antisense approach works for this disease indication. So there's a lot of novelty there, and usually with novelty comes perhaps more challenges than are anticipated when the idea is first thought up. Dyne Therapeutics is also using a transferrin monoclonal antibody. In this case, they're delivering antisense oligonucleotide payload, which I believe is a phosphorothioate. They're delivering a phosphorothioate oligonucleotide to inhibit the expression of DMPK. I hope in the course of the last couple of slides I've demonstrated pretty clearly why an siRNA approach is probably more potent and actually even safer than an antisense or a phosphorothioate oligonucleotide approach. A brief discussion of our muscle atrophy program. Our muscle atrophy program is targeting a disease which can be common or can be rare, so we can use this program either to target common diseases like cachexia and muscle loss and related to immobility, or you can potentially use this program to treat muscle loss in rare muscle diseases as well. Most of the industry, and people are aware of this, most of the industry has been looking at protein loss or muscle degradation by trying to increase protein synthesis by going through a pathway associated with myostatin or the myostatin inhibitor, where they can get muscles to grow. On the other hand, there's this whole other side of the seesaw that's demonstrated here, where protein degradation also increases. And so if you can modulate both sides of this equation, then you may be able to have a more effective way of modulating the biology associated with muscle atrophy. There's one gene that we're targeting, which is called MRF1. It is a E3 ligase. It happens to be at the nexus of the inflammatory cascade as well as in the growth factor cascade. It's a good target to go after, and pharma has been trying to go after it with small molecule inhibitors of MRF1, but because it's a member of the E3 ligase superfamily, it's been very difficult to come up with specific inhibitors with MRF1. Here we can take advantage of the selectivity of an siRNA molecule and create an siRNA molecule based upon the sequence of MRF1 that specifically inhibits that gene and that gene alone. And we've done that, in fact. And if you administer on slide 24 that siRNA to a mouse, in this case, again, using an intravenous administration, this is a mouse whose leg had been denervated, and you can see in the black squares the loss of muscle associated with that. And then in the blue squares, what you can see is that with treatment, there's a 54% sparing of muscle loss. And on the right side is simply shown the gastrocnemic weights, and you can see again there's a significant sparing. Interestingly, this particular siRNA is extremely potent in the mouse. And a single dose of 3 milligrams per kilogram showed on slide 25 actually produces knockdown well past 20 weeks. So this is an example, again, much like in Inclisiran, where you might be able to dose a drug against MRF1 for muscle atrophy and have relatively long-lived activity. Our muscle atrophy program is moving forward. You can see the boxes that we've checked. We expect to begin our first clinical trials in 2022. The next program I'd like to discuss with you on slide 28 is our program in Duchenne muscular dystrophy. Duchenne, as you all know, is related to a mutations in a gene called dystrophin. When there are genes that lead to an out-of-frame reading of the messenger RNA, you don't get any dystrophin protein formed, and that produces muscle toxicity because the dystrophin protein acts as a shock absorber and prevents muscle tearing. Without that protein, muscle cells tear every time you use them. So over time, boys with this disease will lose their muscle function because they simply tear the muscle cells. They eventually run out of stem cells to replace the damaged muscle cells. We at Avidity, as you probably are aware of, there are a number of different approaches that are being taken in Duchenne muscular dystrophy. We at Avidity are going after exons 51, 45, and 44. The concept here is that if you can skip over the exons that lead to a misreading of the mRNA, you can get a shortened but functional form of the dystrophin protein. And so what we're doing is attempting to do that. That, of course, is the mechanism action of the Sarepta drugs. What you're looking at here are data from an MDX mouse model of muscular dystrophy. In that particular mouse model, there is a premature stop codon on exon 23. So here we're making a PMO that will skip over exon 23 and allow for the production of the full-length dystrophin mRNA. And in this particular study, a single dose of 8 milligrams per kilogram produced almost no skipping. So you can think of the drug that's labeled PMO, the blue bar that's labeled PMO, as being the mouse equivalent of one of the Sarepta drugs. When you take that same dose of the PMO, conjugate it to a monoclonal antibody against the transferrin receptor, you get a 50-fold increase in the amount of dystrophin that's produced, suggesting that this is a very efficient way to make a drug which had relatively low efficacy and low potency into a much more potent and much more efficacious drug. In fact, in doing so, on slide 31, we've done a series of experiments where we've given different doses of the oligonucleotide. And in this particular graph, what you're looking at here is the percent skipped, so how much of the right product we're now making versus the concentration of the PMO in each one of the individual biopsies that's represented by each one of those points. And what you can see is that once you reach concentration in tissue that's slightly greater than 50, you don't get much more bang for the buck. So we've actually saturated for the first time the skipping mechanism, and I've worked in the oligonucleotide space with muscular dystrophy for 15 years, and I've not seen anyone be able to saturate it. Interestingly, if you give a non-specific monoclonal antibody that has the same PMO bound to it, you can see that we get lots of drug in non-specifically, but it doesn't produce very much skipping at all. So again, you need to have the specific receptor, the specific trafficking in order to get good uptake. And on slide 32 is simply a demonstration that we have some lead sequences for exon 44, and those are moving into development at this point. Again, clinical trials are expected in 2022. I'm going to go through FSHD and Pompe disease relatively fast. In FSHD, this is, of course, is a disease that is devastating. It is slowly progressing. It's caused by an inappropriate expression of an embryonic gene called DUX4. It's actually a homeobox gene that's generally only expressed in the developing embryo, and then to some lesser extent in testis. The overexpression or the inappropriate expression of DUX4 in this disease, since DUX4 is a transcription factor, it leads to the expression of a number of different gene products that should not be expressed in adult muscle, and that leads to muscle toxicity. So it's characterized by progressive muscle loss. It is focal in nature, it's a kind of disease that would really be well served by single injections of a therapeutic agent that would last for long periods of time. And so the approach that we're taking here is we're making an siRNA against the DUX4, the inappropriately expressed DUX4. And you can imagine that in this case, you can knock down DUX4 everywhere except for the testes without any biological effects, and oligonucleotides and antibodies in particular don't get into the testis. We have a number of preclinical studies ongoing, and these programs are actually rapidly advancing to decision points at this point in time. In Pompe disease is a disease where there are effective therapies. They're not effective enough. So the effective therapies for Pompe disease, which is an enzyme loss, is a loss of GAA enzyme, which ultimately results in the buildup of glycogen in body cells. Unfortunately, the enzyme replacement therapy doesn't work very well in skeletal muscle or cardiac muscle. So skeletal muscle and cardiac muscle continue to accumulate glycogen and have glycogen-induced toxicities. And what happens there is that in our particular protocol, what we're suggesting to do is we're suggesting to limit the amount of glycogen that can be formed in these muscles by inhibiting a muscle-specific form of glycogen synthetase. We're not inhibiting the glycogen synthetase that's in the liver. We're only going to inhibit the glycogen synthetase that's in skeletal muscle and cardiac muscle, and that should result in a reduction in glycogen storage in those tissues and the therapeutic benefit. Again, we're in the final research stages there. We've identified the appropriate compounds that we are now optimizing. Just shifting gears quickly, I'm going to talk a little bit about our immunology programs. And in immunology, what we've been able to do is something that immunologists have dreamed of for years. That is, by taking advantage of different cell surface receptors and monoclonal antibodies to those different cell surface receptors, we can now have the ability to begin targeting gene expression in specific subtypes of lymphocytes. So what you can see here is that on slide 41, we have selective knockdown or selective biology in CD8 positive cells, CD4 positive cells, and tumor-infiltrating lymphocytes in activated macrophages. We're particularly enthusiastic about a program where we can change the expression in T regulatory cells because, of course, T regulatory cells are extraordinarily important in immuno-oncology. So in immuno-oncology, of course, you want to downregulate the function of T regulatory cells. But, of course, in autoimmunity, you might want to upregulate the activity of T regulatory cells. So by being able to deliver specific siRNA payloads to T regulatory cells in different disease states, we have the ability to modulate gene expression in T regulatory cells. Slide 42, I'd just like to go over these data because it's also an interesting application in immuno-oncology. This is an in vivo experiment. We took mice that were bearing syngeneic tumors, mice tumors, and we treated them with a transferrin monoclonal antibody, again with an siRNA against SHP2. And what you can see in this slide on the left-hand panel is that in the tumor-infiltrating lymphocytes, we get significant knockdown of SHP2. We don't get any in the control cells. But if you take the spleens from those same mice and isolate the B and T cells, there's no knockdown. So the only lymphocytes where we're going to see changes in this particular case are those that are at the active site. So now imagine applying this to an immuno-oncology setting in which you don't want to have a generalized immune response. You only want to activate the immune system within the tumor. And you can see the potential benefit of being able to take advantage of the fact that only activated B and T cells overexpress the transferrin receptor, and only activated B and T cells will actually have the pharmacologic effect. So these were the kind of data that Eli Lilly saw in our shop a year or two ago and became really excited. And this was actually some of the basis for Lilly establishing a collaboration with us. And with that, I will turn it over to Joe to talk a little bit about the collaboration and our intellectual property platform.
Great, thanks, Art. Moving on to slide 43, the Eli Lilly collaboration is a great example of how we're able to accelerate and expand the use of our technology into therapeutic areas beyond muscle. We entered into this collaboration a little over a year ago. It's focused on immunology, and we work with our Lilly counterparts in research to progress 6 targets to the product stage. Afterwards, Lilly's responsible for further development and commercialization. They pay for our research FTEs as well as development, regulatory, and commercial milestones, and pay us a royalty on net sales. The real benefit to this collaboration is that we're able to apply our learnings to our wholly owned immunology programs, which is essentially the universe of immunology targets out there minus these 6 that are covered by the collaboration. I'd like to focus on our IP estate. We have IP protection on our platform, which covers the antibodies as well as the oligonucleotide chemical structures and their sequences. And we have additional patent protection on the AOC structure and the way that the molecules are arranged in space. And we also have methods for manufacturing AOCs as well. And what these patents do is they provide us protection from Me Too competition into the late 2030s, early 2040s. And of course, the products that arise from the technology platform will have their own composition of matter claims as well. The other benefit to discovering this technology all in-house is that we don't have a royalty stack or royalty burden to third parties to enable us with this technology. Sarah, I'll turn it back to you.
Thanks, Joe. Moving to slide 46, which shows you how our investor base has evolved and expanded over the course of our Series A, B, and C, with some investors like Equal1 being with us from the very beginning, and a large group of investors coming in as part of our Series C. I know many of you will know many of the people at these various funds. Moving to slide 47, which gives you the perspective on some of our upcoming milestones. Certainly we have a very clear focus on our muscle franchise and looking forward to initiating first-in-human studies in our DM1 program next year and initiating first-in-human studies in DMD and muscle atrophy in 2022. Moving to slide 48 to give you the overall view of where we've gone through over the course of this presentation. I hope we've very clearly outlined both our longer-term focus in terms of looking at being able to take our technology to multiple therapeutic areas, including cardiomyopathies, immuno-oncology, and immunology, as well as our nearer-term opportunity within our muscle franchise, and our immediate focus on our lead program in myotonic dystrophy. With that, I'll close out the presentation, sort of say, how excited we are with regards to the opportunities that we have in front of us in relation to our AOC technology, and hopefully being able to deliver oligonucleotide therapies to patients who previously would not have been able to receive them through targeting areas like muscle, cardiomyopathies, immunology, and immuno-oncology. Thank you.